Hello, my name is Navid Ghaderi and I'm a third-year neuroscience student. My research is focused on the development and application of a novel technique to collect brain cells from laser interstitial thermal therapy equipment and extract their DNA with the prospect of identifying somatic mutations that cause epilepsy. Before we move on, it is important to understand what somatic mutations are and what role they play in epilepsy. Essentially, somatic mutations are genetic mutations of body cells that are not inherited, but rather occur during cell division in early development before birth. These mutations are significant because they have been identified as a cause of epilepsy. A current obstacle to somatic mutation identification is the access to brain DNA, which is why novel methods are required to harvest brain cells. This brings us to laser interstitial thermal therapy, or LITT for short. LITT is a new and minimally invasive surgical approach to treat epilepsy that has been increasing in use. The procedure involves the use of a thin metal rod called an obturator to prepare the trajectory of a laser probe covered in a plastic sheath. Due to the nature of the surgery, no tissue is harvested or extracted. However, the surgical instruments may contain residual brain matter after use. Recently, our lab has developed a novel technique to harvest brain cells and obtain somatic DNA from purified neuronal nuclei that remain on depth EEG electrodes after removal, which are another type of equipment that come in contact with the brain. Our objectives with this project was to apply this novel technique to the obturator and laser sheath from LITT surgeries, potentially creating a new avenue to obtain brain DNA and identify somatic mutations. First, we collected the obturator and laser sheath after surgery. Unfortunately, this could not be done immediately as the obturator in particular needs to be kept at hand in case a second trajectory is necessary. We then moved on to nuclei isolation, where we destroy the cell membrane to access the nuclei within it. FANS, or fluorescence-activated nuclei sorting, was used to separate the brain nuclei from other nuclei present, such as bone or skin, while also quantifying the brain nuclei we collected. Next, we specifically took the neuronal nuclei and performed a DNA amplification protocol to increase the DNA yield of the samples, as the original amount would be too small to work with. As such, we also needed to perform a purification on the sample using magnetic beads to make sure we were only left with DNA. Finally, the concentration of the DNA was measured using a qubit assay. If the concentration was high enough, we would use a microsatellite panel to assess the quality of the DNA to determine if we should proceed with the sample and analyze the DNA. I was able to perform this protocol on 4 obturators and 4 laser sheaths from 3 different patients, as unfortunately one of the patients needed to undergo the surgery twice. From the table, we can see that neuronal nuclei were present in both the obturator and laser sheath. We split each of these samples into aliquots that contain 250 to 500 neuronal nuclei each before proceeding with amplification and purification of DNA. Unfortunately, the results were less promising as most samples did not reach the minimum concentration of 13 nanograms per microliter we needed to accurately analyze the DNA. However, 4 samples did meet this standard. But after performing the microsatellite panel, the results indicated that there were no markers in the 3 highlighted samples, and thus the DNA was just randomly put together and did not match human DNA. Unfortunately, the final remaining sample failed as well, as the microsatellite panel indicated contamination. Despite the results, we have confirmed that neuronal nuclei can indeed be harvested from LITT surgical equipment. The presence of these nuclei indicate that this method is still a viable option to consider exploring as a potential avenue for identifying somatic mutations. We have also pinpointed the most likely failure of the DNA amplification to the low quality of cells extracted from the LITT equipment. As previously mentioned, during the LITT procedure, we cannot immediately collect the obturator as the neurosurgeon may need it again, and so keeps it to the side, which most likely causes cell degradation from exposure to air and out-of-body conditions. We are hopeful that the future strategies of cell collection may address these issues. Preliminary strategies include using a second obturator during the procedure in case a second trajectory is needed, or putting the original obturator in a sterile fluid between actual uses and then collecting the fluid with the obturator and brain cells afterwards. With that, I would like to thank my supervisor, Dr. Carl Klein, for all his help and guidance throughout the project, along with my other members of the lab. I would also like to thank NSERC for funding my research and making this project possible.